Good morning, friends. Today I'll be discussing the topic vaccines. And we start with the historical picture of vaccination in the early 17th century, when the wonderful effects of inoculation, of the new inoculation for smallpox, was discovered by the scientist Dr. Jenner. This topic on vaccines I'll be dealing with in the following areas. One is, what are vaccines? What are the basic requirements of a vaccine? What are the different types of the vaccines and the diseases which it prevents? The mechanism of action of vaccines, and lastly, the stages of development of a vaccine. Now, as you all know, Edward Jenner and Louis Pasteur were the pioneers who developed the smallpox and the rabies vaccine in the early 18th and 19th century. So a proper understanding of the immune mechanism, however, occurred only by the 20th century. So this is a picture of Dr. Jenner and the cowpox vaccine. Now, what are vaccines? How do we develop them? Now, the host organism needs from a pathogen time, often days, to mount an immune response against a new antigen. Now, memory cells will permit a rapid response to pathogens which are previously encountered. Active immunity can also be generated artificially through vaccination. So, what's the principle behind vaccination or immunization? Is one, to introduce an antigen from a pathogen. This will stimulate the immune system. It will develop a specific immunity against that pathogen, and no disease associated with that organism is caused. So, what are vaccines? Vaccines are attenuated or killed organisms. They could be antigenic components of disease-causing microbes. They could be toxins produced by the bacteria, or they could be DNA coding for a particular antigen. Now, this is deliberately administered to an individual to protect him or her from a host of infectious diseases. Now, vaccines are a powerful means to prevent and eradicate disease, and the discovery of vaccines is one of the most powerful milestones in medical history. Now, what would you think is an ideal vaccine? The ideal vaccine should be able to mimic the immunological stimulus which is associated with natural infection. Now, there are certain conditions whereby a vaccine becomes an ideal vaccine. That is, it should have no side effects, it should be readily available, it should be affordable, and it should be easily administered. The vaccine should also have certain immunological requirements. So it should satisfy these immunological requirements. What is it? It should be able to activate the antigen presenting cells. It should initiate antigen processing, production of cytokines, the activation of B and T cells, and the formation of neutralizing antibodies. Should be also able to develop memory cells to resist further infection. Now, what are the types of vaccines that we have available? One is the inactivated or killed vaccine. How does it act? They are pathogens which have been rendered harmless by treatment with chemicals or heat. The disease prevented include cholera, pertussis, typhoid, and polio. Now, when we look at the advantages or disadvantage of the vaccine, we see that it's a safe vaccine, but it fails to evoke immune response during natural infection. The second type of vaccine is the live attenuated vaccine. It is attenuated to decrease its virulency, since it's a live virus. So how do you decrease virulency? This is done by repeatedly growing it in culture medium or by recombinant DNA technology, where segment of the gene for virulency is deleted or mutated. The disease prevented by such live attenuated vaccines are BCG, polio, measles, mumps, and rubella. The advantage is that it does not produce disease, it's highly immunogenic, but the disadvantage is that it can induce disease in an immunocompromised host. The third type of vaccine is the toxoid vaccine. Toxins released by the viruses are purified and inactivated and mixed with stable adjuvants, and they become effective vaccines. An example of this is the tetanus toxoid vaccine and DPT triple vaccine. The fourth type of vaccine is known as the subunit vaccine, where you have the identification of specific antigens and the use of these antigens. This has been used in the case of hepatitis B, and it is safe and protective. Now we talked about DNA vaccines. Now, what is the importance of this DNA vaccines and how are they produced? So we identify and culture the organism. We sequence the DNA and we identify certain immunogenic peptide motifs. We identify the corresponding DNA sequences which codes for these immunogenic peptides. Then we construct a plasmid vector which is able to express this immunogenic peptide motifs. The plasmid DNA is further purified and it is used in animals, that is, it's administered as a DNA vaccine to test for its efficacy. Now, the DNA vaccines are at present at an experimental stage, but it holds promise for future therapy since they evoke both humoral and cell-mediated immunity without the dangers associated with the live virus vaccines. Now, what are the different stages of producing the vaccine? First is we have to select the strains for vaccine production. Then we have to grow that particular microorganism. Then we isolate and purify it, inactivate the organisms, and then it goes through a procedure known as the formulation of vaccine. That is, once the purification is done, then suitable bulking agent or an adjuvant, stabilizer, preservatives are all added, and that step is known as the formulation of the vaccine. And this is followed by the release of this vaccine. That is, it goes through the manufacturing process. That is, the vaccines are taken in its containers, and then it goes through the process of quality control. And once the quality control is done and the labeling has been done, it is suitably packaged and it is then released. What is the mechanism of action of vaccines? Now, a vaccine is a pathogen mimicking molecule. Now, this pathogen mimicking molecule or the vaccine is then captured by the antigen presenting cells or the APC cells, and it activates both the immune and T and B cells and produces the antigens, which then produce antigens against the it produces antibodies against the antigen. So this in turn will now activate the memory T and B cells so that subsequent attack by pathogen becomes stronger and we have the generation of herd immunity. So there are two main stages in the development of a vaccine. One is the preclinical stage, which I've shown you earlier. That is, it has to go through the good laboratory practice guidelines, that is, which I showed you in my previous slide, where all measures should be taken in its production, characterization, and quality assurance of the candidate vaccine. It is then tested for toxicity and safety. Its potency is tested, and so also its immunogenicity, and suitable, as I told you, while it is formulated, you have to add the suitable adjuvants and additives. Once the vaccine is released from the laboratory, it has to go through 3 main phases of a vaccine trial. Phase 1 is where we test the safety and immunogenicity. Phase 2, we test both the safety and we also test the dose where it is effective. Dose, the range at which this vaccine would be effective. And in phase 3, which is a large scale population, we look at again the safety and the efficacy of our study. So let's walk through the different stages in the development of a vaccine or in the creation of a vaccine. First, there is an outbreak of a pandemic. So there should be a robust surveillance and monitoring system. The epidemiology of the disease has to be studied. What is the disease burden and how is it affecting the health economics? All this is studied before we go into the process of preparing a vaccine against that particular microorganism or a virus. The next phase is known as the preclinical development. In the preclinical development, we create the antigen, it goes through the process of purification. We study this in relevant animal models. We check its toxicity and we goes through certain clinical trials and goes through regulatory submission for these clinical trials. So the preclinical phase involves mainly the purification and preparation of the vaccine at the laboratory level. Okay, where we prepare the vaccine, you formulate it, you test it goes through quality checks. We do some clinical trials, maybe on animal models also, and we check the relevance of the vaccine and its potency. Next, it goes through the phase of clinical development. Now, as I told you, there are 3 main phases. Phase 1 checks the safety and immunogenicity in a small number of population. Phase 2, the safety and efficacy is done in hundreds and thousands of volunteers. And phase 3 becomes a multi-center trial where the efficacy, safety and immunogenicity is done on a very large number of subjects. So phase 4, we look at the continuous monitoring of adverse events and any new indications that we observe. And finally, the certification is done by regulatory bodies. And the last phase is the manufacturing phase, where it then goes for large-scale production of the final vaccine, and long-term storage validation is done. How long can it be stored? What are the precautions to be taken? And the precautions taken to be transported to the rural and urban areas. So these are the various stages for the creation or the development of a vaccine. First, we have an outbreak of a pandemic which is studied very closely, and then we go into the preclinical stage, the clinical stage, and the manufacturing stage comes right at the end, where all the 3 phases it has gone through the 3 phases, and the 4th phase also, where we monitor any adverse events, and they are all observed and noted down and corrections made, and then certification is done by the regulatory bodies before it can go to the final stage of development and production of the vaccine. And very importantly, what we have to study is how long it can be stored and how it can be transported to the rural and urban areas. Now, summarizing these aspects, the early vaccines were mostly dead or live attenuated. These provide their own integrated adjuvant or antigens. Now the shift is towards defined subunit based vaccines. This requires us to select the adjuvant and the antigen to be included. So immunology is now having a greater impact on vaccine, and the key for a good vaccine is its safety, its efficacy, and the memory. Now, as you all know, Corona is now the burning area for vaccine development, and the race to the vaccine is on. More than 140 coronavirus vaccines are under development, and around 15 vaccines have gone through phase 1, testing for safety and dosage. 10 of them have gone for phase 2 trials, that is, they have been expanded for, that is, in a larger population, they are looking into safety trials. 4 of them have gone through phase 3 trials, that is, in a very large population, multi-center, very large populations, its efficacy is tested. And only one vaccine has shown some limited use, has been approved for limited use, but this is also under question. Okay, so this broadly covers the topic on vaccines, what they are, and their various stages of development, and also I have told you about what is an ideal vaccine and how it should be prepared. Thank you.